awesome to be talking about the Ecological Researchers Education Network. I'm going to go ahead and share my screen. So, welcome everyone, and here's the plan for today. I'm going to give an introduction to the Ecological Researchers Education Network, and I'll be doing that in my role as past president of the board of directors for EREN. Then we're going to have presentations by 3 EREN lead scientists who will describe the projects that they're doing within the network. And then we'll have questions and prompted discussion. We were thinking of doing breakout rooms, but given that we don't have a huge number of attendees, we're just going to stay together in the main room at the end and have questions. And if things start slowing down, I'm going to share a couple of prompts that I thought might be fun for us to discuss. And we will end at 4 o'clock. So that is the plan. So just to describe what EREN is for anyone who is not familiar, we were founded by a grant from the NSF Coordination Network Undergraduate Biology Education Program. And we were funded from 2010 through 2016, but we have persisted to the present day, which is very exciting. And we have done a number of things over the course of our tenure as a research coordination network. We've gotten 2 additional NSF grants to work within EREN to develop some projects with some special focus, and I'll talk a little bit more about that as we go along. Our main mission is to create, facilitate, and promote collaborative ecological research that generates high-quality, publishable data and involves undergraduate students and faculty, and our kind of target institutions are primarily undergraduate institutions. But really, anyone is welcome to join EREN if you have a project that kind of fits with our project model that I'll explain in just a moment. And EREN is really working to bring what we call big science to small institutions. We're thinking about institutions that are under-resourced or faculty that are under-resourced within their institution. And we're imagining ways to kind of spread our science across multiple sites. The idea is really thinking about the fact that when we teach ecology, all of us are taking students out to the field, and EREN is really built on the principle of what if instead of just doing all of these independent projects, we coordinated our efforts so that all of the students were working on the same project, all collecting the same kind of data across many sites, and we could put that data together and maybe see some really interesting large-scale patterns. And we believe that this would enhance both the scientific and educational value of the things that we're doing with our students, because they're no longer just doing sort of a token data collection, they're contributing to a project. And this could go on over multiple years, once again over multiple sites, and it makes for a really exciting thing for both the faculty and students to be involved in. We have a lot of members who have joined EREN over the years, over our 15 years. My last count was 470 members from over 300 institutions, and these are all, approximately marking the institutions. We also have people from different countries who have joined EREN. So it is a large network, and all of these folks are interested in collaboration. So it's all based really on the projects. We invite our members to propose project ideas to the network through the EREN website. Members volunteer to join projects of interest. Excuse me. Each project organizes under a lead scientist to develop protocols, collect data, and seek funding. And often there are teams of lead scientists. I want to make clear that often there are 2 or 3 people leading the EREN project. And what EREN does is really facilitates opportunities to communicate, share data, post information, and meet face to face. And we also host, this network of people that are interested in collaboration and you can reach out to if you're looking for collaborators. Our projects, excuse me, focus on some common themes. They are diverse, but they have some commonalities. They all focus on ecological or environmental science. They involve coordinated data collection across sites. They involve undergraduate researchers. They strive to contribute to peer-reviewed science. They strive to educate students and faculty through immersion in collaborative research. And they really strive to be accessible to all institutions and researchers. Here are a few of the projects that we have hosted over the years. We have one that looked at stream temperatures across a large environmental gradient. And this project is now complete. We have a manuscript from that. The Permanent Forest Plot project is one of our most popular projects with about 60 sites contributing. And we're trying to publish a data set from the Permanent Forest Plot project. And we'd like to acknowledge scientists, including Dr. EREN Lindquist, who have worked on that, and the current lead scientist, Dr. Karen Coors. We also had a project called Turtle Pop, and this was looking at turtle sex ratios across a range of ponds and a range of habitats across the eastern and midwestern U.S. That project is now complete, and we now have Turtle Pop 2.0 underway, and we'll hear a little bit about that project in this session. We also had a project looking at decomposition of terrestrial and aquatic invasive species across a range of sites. A manuscript came out from that project, and we appreciate the work of Carolyn Thomas on that particular project in the past. Now, we were working on these projects very successfully, but I do want to mention that we had some selective pressures upon us. We had the COVID-19 pandemic. We had more and more environmental data coming out. We also had the National Ecological Observatory Network developing right alongside EREN over the past 10 and 15 years. We also had macrosystems ecology emerging. And also, we all recognize the need for to be welcoming to a range of folks. So some of our more recent projects kind of spin off of some of these selective pressures, if you will. During the COVID-19 pandemic, we had a project with NEON that was funded by NSF with emergency response funds to quickly develop projects that could be done by students collecting data pretty much anywhere, not necessarily on campus, but in their backyards and, in natural areas nearby. And those projects are still available on the EREN website for flexible learning. They're called EREN Neon Flexible Learning Projects. We also partnered with NEON and Project EDI to really try to embrace some of the large data sets that have been becoming available to us, and recognizing that EREN has this framework for designing collaborative research projects that's really great, but Project EDI has developed a lot of techniques for teaching data science, and of course NEON has these wonderful data sets that we can use. We combine those all together into a grant to try to create projects that have more of a data focus embedded in them as well. And these are the EREN Neon EDI projects that have emerged more recently, and you'll hear about some of those as well. Our latest endeavor is called the Macrosystems Ecology for All Network, or MIFA, and this is also getting into, looking into macrosystems ecology and data science. and trying to combine that with our collaborative, the collaborative spirit that EREN has worked on for so many years. This is actually a different network, and I won't talk too much more about it, but it is something that grew out of EREN, and a lot of people working within EREN are also working on MIFA type projects and on leading the MIFA network at this particular time. So that's a grant that's currently underway. A final new development is that we have a new president for the EREN board. Dr. David Bown has just assumed the presidentship, has been elected this past summer, and he's a professor at Elizabethtown College in Pennsylvania. He's a founding member of EREN. He was the lead scientist of the first iteration of Turtle Pop. He writes really cool ecological fiction and is an interdisciplinary collaborator. And I'm thrilled to be handing the EREN board off to him. So that's another new development that we have. And that kind of takes us to the end of my presentation. I just want to summarize that we have sponsored 19 collaborative research projects. We have 470 members from over 300 institutions who are interested in being collaborators. We have 15 years of operation. We've produced 14 multi-author papers. We've involved thousands of undergraduate students in authentic research through the EREN projects. And you're welcome to join EREN for free. There is a QR code if you'd like to go straight to the EREN website. And hopefully you will join us. And I'm going to stop sharing my screen. And I'll go ahead and introduce Jennifer Purenhage, Dr. Jennifer Purenhage, who will talk about an example EREN project. And you can see through a direct example a little bit more about how the EREN projects work. So go ahead, Jen.
So I'm Jennifer Purenhage. I'm at the University of New Hampshire, and I am one of the co-leads of EREN's Turtle Pop 2.0 project. My two other co-leads are Dave Steinberg, also here at UNH, and Ali DeGrassi, who's at Shenandoah University in Virginia. And I'll just mention that, as Lori said, EREN began as and is largely folks from PUIs. Dave and I are both at UNH at an R1 research institution, but we're both in teaching positions that have very heavy teaching loads, and any of our research is really with undergraduates. And so that involvement in EREN has been great for our research and for including our research into our teaching. As it is, of course, for Ali and other participants of the project. The last slide was there to remind me to explain why we're 2.0, but Lori already kind of introduced the idea that there was a Turtle Pop, an original Turtle Pop, and that was led by Dave Bown, who's our current EREN president, and I believe is here today. And I personally got really interested in EREN and joining EREN because of Turtle Pop, and I really wanted to be a part of Turtle Pop. And I came in right when Turtle Pop was ending. And so reached out to Dave and asked if he was going to continue leading the project. He had said he was going to step away from it for a bit, but that he was really encouraging of us reviving it and gave us some suggestions about how we might adjust the design based on their experience with Turtle Pop 1.0. So we took that advice, and we are trying right now with this iteration of Turtle Pop to use a paired ponds design. So to still ask this question about how urbanization affects turtle population abundance and population structure, sex ratio and age structure. But to do it not just by collecting ponds anywhere along the gradient, but to try to actually pair urban and rural sites, and also to increase our sampling effort. And so we've done that by using smaller, less expensive traps and using more of them. And so we're still asking the same overarching question of, how do urban and rural painted turtle populations differ in terms of population size and structure, but with a slightly modified design. I wanted to mention here at the top that in our only few years in doing this iteration of Turtle Pop, we've had hundreds of undergrads already at about 14 different colleges and universities participate in Turtle Pop 2.0 through their courses, lots of different types of courses, through independent research and through extracurricular opportunities. And I wanted to share, I had some of my own pictures, but I replaced them with these photos and this text exchange that I had with our co-lead, Allie, just yesterday. She reached out to let us know that after taking a year off from turtle trapping, they were back, and how appreciative she is for being part of this project and working with turtles. She's a small mammal ecologist, unlike me and Dave, who are herpetologists. And she was just saying how much her students love the project, how much it adds to their class, and how happy everyone is. And I wanted to share that too. Anyone who's worked with turtles knows that it's hard not to smile when you're holding a turtle. So really encourage you to get involved, even if you've never worked with turtles before. So what does it take to participate? It takes a pond, a rural or urban, or both if you have them, that has painted turtles in it, certain permissions that we can help give guidance on getting, and about $300 to $350 worth of equipment and supplies, some of which you may already have, and we have that all clearly laid out for you in our materials. You also need to be able to do 2 trapping events per year in August or September. Sometimes that extends to October, depending on where you are in the country. And that's it. That's all you need to participate, and some willing people. And so what does it look like? You show up in the field, you have 10 of these small minnow traps that we use now at each site, and you record water and air temperature, and then remove all the turtles from the traps and identify them. And for at least painted turtles, take 6 different morphological measurements per turtle. Some of those measurements help us understand their approximate age or life stage. And some of them help us sex the turtles, which we do for every adult. And then we also use capture, mark, recapture to estimate turtle population size. And so we mark every turtle with a notch code on their scutes, on their carapace. And it's a non-invasive, relatively non-invasive method, and really easy to do. And so then what we offer as a project to all participants is a detailed protocol with a photo guide showing you exactly how to do every measurement and every step of the process. Of course, data sheets and notch codes, language to help you get permission to do this work with vertebrates, training videos. We have a YouTube channel that we're happy to create special videos for if you want us to train you on something. And we already have a few up there. And of course, teaching materials. We're happy to share any of our teaching materials. And we are currently accepting new participants and happy to answer any of your questions. We're about 3 years into data collection on this new version of the project. You don't have to participate every single year, and we would love to have you. So thank you very much.
Thanks so much, Jennifer. All right. Now I'd like to introduce Mary Beth Kolasvari, and she's going to be talking about the vernal pool phenology project.
Awesome, thank you. I'll be presenting on the vernal pool phenology project, and our overarching question is, how do local and regional factors affect the spring phenology of pool breeding amphibians and trees? We piloted this study in 2022 and then launched the full project in 2023, where we've been collecting data every year. In the eastern US, vernal pools are small temporary ponds embedded within forests. They provide important habitats for a variety of forest species, including key breeding habitats for 2 of our focal species for this project, wood frogs and spotted salamanders. Wood frogs and spotted salamanders use these pools for egg and larval development, but the remainder of the year they disperse throughout the surrounding forest. Thus, vernal pools are hotspots of biological activity, and energy flows from vernal pools to the surrounding forested landscape. These pools dry regularly, so they lack fish predators, so they're predator-reduced environments compared to permanent ponds. Both wood frogs and spotted salamanders in many vernal pools breed in very high numbers, and in many parts of their range, vernal pools are critical for sustaining populations of these 2 species. Hydroperiod is particularly important for productive vernal pools in that they need to hold water long enough for amphibian larvae to reach metamorphosis. In our project, we also look at dominant trees surrounding the pools. So overstory trees that are immediately abutting the vernal pools, think about it. They're shading the pools, affecting water temperature. Deciduous trees provide leaf litter and other debris inputs that fuel vernal pool food webs. And when trees begin to put energy into seasonal growth and reproduction, they take more water and have the potential to affect pool hydroperiod. So thinking about how these ecosystems are the potential for climate change and how that can affect these ecosystems and how that as they interact with the surrounding forest. So as climate changes, are all members of the community changing at the same rate in the same direction? Will climate change affect the ability of these pools to continue to hold water long enough for successful reproduction for these key species? Are there phenological mismatches of members of the community that may affect the sustainability of these systems and all their components into the future? So what do you need to participate in this project? You need a vernal pool surrounded by trees. The pool can be temporary or permanent as long as it lacks fish. It also has to have, you have to know that at least one of those focal species, wood frogs or spotted salamanders, breed at the site. In terms of equipment, costs are relatively low. You would need hobo temperature data, like data loggers, about 2 of them, which are roughly $70 each. Acoustic recorder, we use the Audio Moth primarily, which is has open source functioning, and it's about $150. We do not want those costs to inhibit your participation. We have collaborators that have access of this equipment that if this is going to potentially prevent you from participating in this project, but you want to, please reach out to us. If we're able to help you and send you equipment, we're more than happy to do that. Water gauges, on the other hand, are less than $10 to make. So usually that's not the limiting factor in participating. You also need to commit to twice weekly surveys during our 6-week sampling period, which is in the spring when everything's happening. In terms of data that we collect through our field surveys and through these data loggers, we'll collect water level fluctuations. We get measurements of canopy cover, maximum depth. Our data loggers that collect air and water temperature give us, in addition to the temperature, they also allow us to determine dry date of the pools. In terms of phenophase data, we're interested in the first day of egg deposition for wood frogs and spotted salamanders. And for wood frogs, we're interested in the first day they begin calling on site, and we can get that through the audio moss. In terms of trees, we're also, those dominant trees surrounding the pools, we're interested in documenting any phenophases that occur during that time period. So the first day of things like bud burst, leaf unfolding, open flowers. It really depends on the species, whether or not you're going to see them or not. There's no need to monitor these tree species after that 6-week time period. So this map here shows the distribution of study sites we have to date across the range of the species. We're interested in more, but as you can see, this is a wide distribution and we really need that large sample size across a broad geographic range to answer our research questions. To date, our partners or participants have incorporated this at colleges and universities into their field lab classes or independent research projects. So that's about 60% of our participants. We've also had a decent number of educational, environmental education centers. About a quarter of our sites are from those types of institutions. We've also had, high school and middle school classes or students participating in this as well. Basically, we'll take anyone that's interested in participating. We're very welcoming. We're still recruiting new participants to this study. Certainly, it would be great to have people that are joining the study be able to monitor that same site multiple years. For many of our analyses, we're going to need multiple years of data on the sites. However, we want to be welcoming and inclusive and increase our sample size, so we offer flexible participation. So at a minimum, you would need 1 year's worth of data as long as you collect all that data during that 6-week sampling period. So here is a QR code for you to get to more information on our project, on our webpage. And if you are interested in participating or have any questions, please email us at the email address provided. We'd love to talk to you more about it.
Thank you so much, Mary Beth. I'm also participating in this project, and it's super fun to do as well. I'd like to introduce our third lead scientist, Elizabeth Sudeth, and she's going to be talking about the INVERTS project.
INVERTS stands for Investigations with Neon, Variability of Macroinvertebrates versus Urban and Rural Temperature Dynamics in Streams. And we're very proud of that. Shannon's on the meeting. Shannon came up with that one. As you can see here, we've got a great picture of some of my students in an urban stream in Atlanta and some of Patty's students in a rural stream in Ohio, and there's some bugs. So we were inspired by that original EREN stream temperature project. The one thing that they mentioned offhand in their paper was, hey, I wonder how this stream temperature variability affects biodiversity. So we decided to investigate that. So we're interested in how the thermal regime, which is the variability in temperature in a stream over time, affects biodiversity. And so the thermal regime is affected by the local climate, by local topography, and it's also, of course, going to be affected by things like land use. And so by thermal regime, we mean variability across lots of scales. So daily, weekly, monthly, seasonally, annually. We don't know exactly which of these variables may matter yet, so we're getting it all. We have formulated 3 hypotheses that we are going to investigate. Our first hypothesis is based on the intermediate disturbance hypothesis, and this is the idea that we expect biodiversity to be higher with a sort of median range of thermal variability compared to more extremes like very little variability or extremes like a whole lot of variability. Our second hypothesis is based on the urban stream syndrome, and this is the idea that continental scale variability we see based on climate and topography starts to disappear as areas get more urban, and urban areas across the world tend to be more similar to each other. And then our final hypothesis, which we are still working on how we're going to test this the best, is based on the thermal equilibrium hypothesis, the idea that for a given species, it has a range of temperature where it will do the best. And we're interested in seeing if we can see signs of that in our data. So when I say us, we have been an EREN pilot project for the past 2 years. And the little airplanes represent our participants to date who have already started collecting data. But we're also, as mentioned in the title, using neon data. And so the little blue symbols there represent all the neon sites across the entire US. And that will allow us to get sites much more pristine than those where any of us work, and also allow us to get sites in areas where we do not yet have any EREN participants. And so especially if you happen to be in the Western US or the Great Plains Midwest area, or Puerto Rico or Alaska or Hawaii, we would love to have you join us, but we'd also love to have you join us from anywhere. We can always use more data. So what do we ask of you? Our whole focus is on thermal regime, so the number one thing we need from all participants is at least 1 year of continuous temperature data. As others have mentioned, temperature loggers have gotten really cheap, so you would need a temperature logger that you could deploy for continuous logging in a local stream, or you can use an existing local gauge. And both of my sites are at USGS gauges. It saves me so much headache. I don't have to worry about maintaining the gauge myself. And my gauges are safe currently from funding cuts. So you need that. We need at least 1 year of data to compare to the rest of your data. And then we have developed a habitat assessment, and we're trying to do that at least once a year, even more at each of our sites, to help us collect all different kinds of physical data and observations about the sites. And then we need biological data. And we have two different kinds of macroinvertebrate sampling. We have protocols for doing traditional macroinvertebrate sampling, and we also have protocols for collecting stream water that is then filtered, and the filters are sent to Shannon in New Hampshire, where she's going to analyze for eDNA. And so another part of this project will be a cool chance to compare morphological sampling versus eDNA sampling. And like the others have said, this project is very welcoming to you, even if you're not positive still what a macroinvertebrate is. We have excellent protocols, and we work really hard to help you get up to speed and feel comfortable doing all the way to doing your own identification. And for this project, we're asking our scientists to do identification to family, and then also save the bugs so that we can ID them further as needed in the future. So we have a very flexible design for you to use at your campus. Obviously, first and foremost, we want you to contribute to our long-term data set and work with us to answer our research questions at multiple scales and help us write these cool papers we're going to write testing these hypotheses. We've written these as modules that you could use just for a single lab in one class. You could use it as multiple labs in a class, or you can even use our project as your semester-long cure project and theme your whole class around it. And we've already had this adapted and used in a range of different kinds of classes, including intro-level bio and environmental science classes, both for majors and non-majors, upper-level classes like ecology, limnology, and conservation biology, and also a lot of folks are doing it as directed research projects. So having individual students spend a semester or more focused on inverts. And so that's our quick and dirty introduction. Feel free to email us at inverts@gmail.com or scan our QR code, and that link will take you straight to our webpage on the EREN webpage, where you can see our protocols and follow a link to get all the information. So if you have a lot of questions about how could I do this, could I do this, you can read all about it and learn a lot, as well as emailing us and asking us all the questions you want. So we are Shannon O'Leary at St. Anselm College, me at Georgia Gwinnett College, and Patty Saunders at Ashland University. Thanks.
So that ends the presentation portion of this water cooler chat. And I'd like to, as I said at the beginning, we thought if we had a lot of people joining the chat, like several dozen, it might be easier to go into breakout rooms. But since we have a reasonable size for conversation, we will stay in this breakout room or in this main room together. And we'd like to invite any questions you might have. You could ask questions of the particular lead scientists if you want to know more about their particular projects. If you have any questions about EREN in general. I will also just mention as one final thought to leave you with, we wanted to hold this water cooler session because we do feel like EREN projects are a great strategy under the current funding situation. Our projects are really designed to be very low cost. I think everyone emphasized that when they presented the projects. And also they're really designed to, kind of build community with academics. Academia is a difficult place to work right now because of the particular climate that we're in. And so we'd like to kind of offer you the EREN projects as a way to go forward, especially if there are issues with funding and so on. But does anyone have any questions that they'd like to ask? You could raise your hand or put something in the chat.
How long do you foresee these projects continuing into the future?
Do you have a particular project you're thinking of?
They all sound cool, but I think I'm probably, if I have to pick one, I would probably choose the last one.
What do you all think for inverts, Elizabeth?
So as I mentioned, we kind of took a long pilot period. So we are just started our first official year. We're expecting to run for at least 3 years.
Other EREN projects, 3 years of data collection is not, that sounds actually kind of typical to me. some go shorter, some have gone, I mean, the permanent forest plot project is kind of still ongoing. But that, of course, is dealing with very long-lived organisms, with trees. So that, that goes a while.
And if I could
Yeah,
jump in
go
there.
ahead. Yeah.
Yeah. Hi everyone. So I'm Dave Bound, the current president of EREN. And as Jen and Laurie mentioned, the previous the first director of the first Turtle Pop. And I just want to add in, one of the great things about EREN is the flexibility of the projects, but also just the question about how long do they last, right? Like in my case, Turtle Pop, we collected data over 2 years and it was successful. And then there with Jennifer and her colleagues taking on 2.0, right? It has a life of its own now, right? So all these projects can outlive kind of the original conceit. We just have to have the people willing to get excited and take them on. So to me, that's the ideal situation, right? You start a project and then the next generation takes it over. So just throw that out there.
I see also that Nick has placed a question in the chat saying, IACUC approvals have been difficult for me in the past. Does the Turtle project provide help or an umbrella permit? That might be for you, Jen. Yep.
Yeah, I just answered in the chat, but I'll just
Okay,
say,
great.
we don't provide an umbrella permit because in any given year, well, there's two different things going on. One is collecting permits in an individual state. And so we can't provide necessarily an umbrella permit for that. And similarly, IACUC, the animal care and use at each institution has to be by that institution, your personal institution's IACUC. And I know that all IACUC committees are not created equal. They're all like, not the same ease of getting through an IACUC committee. I've seen that at different institutions. But we do provide our language, our specific language that we used to get our IACUC approved and to get our fish and game permit. And so we have all of that information available in our project participants folder, so that they can use that, and hopefully that will get you partway there. And we're always happy to help if your IACUC pushes back, and to try to, give help on that. So yeah,
Great,
hopefully that
thanks.
answers the question.
Yeah, that's perfect. Thank you.
Yeah.
There's another question in the chat from David Ando. How does a project initiator recruit others to participate, and how long does it take to acquire participants? And that's a great question. We have a little bit of information on the EREN project website that says how to propose a new project. So if you have a brand new project idea that you think would fit well into the EREN model, there's a procedure that we follow that's on the EREN website. And our usual model in the past has been, once the project proposal has been accepted by the EREN board, then we have the project leader or leaders prepare an email that we circulate to the EREN email list. And we encourage you to put the information that our project leaders have shared, things like how much will it cost in terms of equipment, what exactly is expected, are there opportunities to be an author on an eventual publication, or is this sort of more pedagogically oriented? Just some of those basics go into kind of a, let's say a 2-paragraph email or a 1-page sort of description of the project. And then people respond directly to the lead scientist. And often you get a core group of participants fairly quickly through the email list as soon as you circulate your proposal. But as you can see, the 3 scientists we have with us today, I mean, they began their projects, and we often do something called a pilot phase where you're like, okay, we're going to try our field methods before we're going to collect data that we call our real data. And often that phase will last 1 to 2 years. And then you can recruit additional participants, once you have your methods worked out. So it varies a lot project by project, but that's been the typical pattern. Does anyone else want to answer that, Mary Beth?
Sure. I'll also add, it really depends on the particular project. So as Lori said, she would do an email blast to EREN members. We advertise these widely on Ecolog, but for example, our particular project, we advertised it across our own networks and the Northeast Partners of Amphibian and Reptile Conservation listserv, as well as the park listserv, and that's how we got a lot of our like folks from like the zoos and aquariums and other places participating. So a lot of that is project specific.
A word of mouth, I think, within the EREN network and just people sharing it with people they think that it would be relevant to. And I think like Mary Beth said, different projects I think will gain participants at different rates. So like depending on the ease of entry into a project, what equipment's required, how easy it is for people who don't have experience with the system, maybe you get more participants quicker than other projects. So yeah, it varies a lot, I would think.
So say we've tended to lean toward projects that are more accessible. In other words, people can be relatively quickly trained to collect reliable data. They may not need deep expertise in that area. But there's some other projects that, and these are fewer, like I said, but sometimes people need more of a time commitment or more expertise is required. And those projects, will have fewer people involved, but that might be fine for the research question that is being pursued. So yeah, it can be a little variable in that regard. There are questions. And feel free to unmute if you'd like as well. Our group is not so large.
I just wanted to point out that I put in the chat, and if you're watching this later, you can look on the EREN website and navigate to this pretty easily, a link to something called EREN
Excuse me.
Projects by the Season, which is a kind of a table that we put together to show you, if you're teaching, which semesters, which times of year it might be good to try to involve your students in different projects. So if you're thinking about course planning, you can look at that and see which projects might line up with your semesters or your trimesters. So I just wanted to let you know that resource is there. It's called EREN Projects by the Season. I put it in the chat.
I'll pipe in and mention one other thing. I believe that many of the projects that maybe have kind of sunsetted in terms of not being an active research project anymore, they've published, but we still host the teaching materials on the website. So just going through the EREN projects, whether or not they're recruiting or not, you can find a lot of excellent resources for teaching. And we know we know what it takes to try to implement and incorporate all these types of exercises into labs. And so we do try to provide as much flexibility and as much guidance and as much information to make that transition to being able to adopt a project as easy as possible. So there's just a lot of different resources out there on the website.
I put a couple of questions that occurred to me as I was thinking about this water cooler chat and thinking in the spirit of, having a little bit of exchange in addition to presenting these EREN projects as opportunities. And thinking about, the questions that I had were, what are the most significant challenges you are facing in terms of engaging undergraduate students in authentic research experiences? I ask that question because I feel like EREN really emerged out of a commitment to, as a teaching faculty person, do authentic research with our students and do it in a way where it might be especially impactful because we were collaborating across multiple colleagues and across multiple sites. And just for informing EREN's work in the future, are there things that networks like EREN might be able to offer that would address these challenges? This is something, Dave Bown and members of the EREN board can be thinking about as we move forward. And I thought I'd just put those out there and see if anyone has any thoughts about either challenges or things that we could offer through our network structure that might be helpful. And I see someone has put something in the chat. Madhura has mentioned fast-changing technology, especially on the computing data science side. This is what MIFA is already trying to address. Yes, I totally agree. I'm feeling like the data science and data opportunities are moving so quickly. I worry about being able to kind of educate my students at the cutting edge and trying to embrace, and I mean, that's something Elizabeth's project, the Inverts project, is doing, is really trying to use the NEON data in partnership with data collection in the field by students and see how those two intersect.
We already have a couple teaching modules sort of aimed at that idea, and it's something we'll continue to work on once we have more participant data to sort of show you how you can use your participant data and NEON data and help your students learn some data skills.
Laurie, can I jump in?
Yeah, please.
Yeah, I just want to, so David put into the chat, one major challenge is having enough possible choices for students to select for a high impact experience. So one of the great things that one of the great things about EREN, I think Mary Beth mentioned this, and I'm just reinforcing, is the website has so much information. So if you don't want to, be an active participant, you can still use the protocols. So I know in my general ecology course, I assigned, every pair of students has to come up with their own independent project. And a lot of these students, they have no idea what they're doing, right? And are just, flounder a little bit. And I say, just peruse the EREN website, see what's interesting to you, and then read some of their stuff. And it's amazing how many students, grab onto that idea. There's one in particular, earthworm distribution. For whatever reason, students find, putting a bunch of hot mustard powder solution in soil and seeing earthworms come up is just really intriguing. And so it's their kind of entryway into ecological research without having to start from nothing. And there's many other examples on EREN. So you can kind of use EREN, without actively joining. And we would love you to actively join with the projects, but the resources are there.
Well, I really hope that you all will think of EREN as you're doing your teaching, as you're designing new research projects. And remember, if you have a project that you think would really benefit from this distributed collaborative approach, EREN is meant to serve the community in that way and be kind of a clearinghouse and be a place where people can connect who want to collaborate. And I really appreciate everybody attending the session. Thanks for the comments in the chat. And we will go ahead and end. So thanks very much. And thank you to Jennifer and Elizabeth and Mary Beth for taking the time to present their projects. It always gets me jazzed again to hear the details of the projects. And I'm like, these are so cool. Even though, of course, I'm familiar with them. So it's great. Much appreciated.